called Andreassen, and I'm going to give a little presentation of bipolar disorder and the clinical characteristics. And bipolar disorder, the diagnostic features includes episodes of elevated mood. This is called mania and includes high activity, little sleep, and kind of high self-esteem. And that is a specific period, and then you have normal mood in between. And in addition, there are episodes of depression, meaning low mood, low activity, little energy. And the bipolar disorder globally affects 1 to 2% of the population. It's a major disease globally. Currently, the treatments are mood stabilizers such as lithium, and the inclusion of psychosocial treatments like education, learning about the disease, and lifestyle measures and cognitive therapies. Currently, the causes of bipolar disorder is not fully known, but there is a high heritability, meaning there is a large effect of genetic susceptibility. But to release or to start the disease, genetic factors, they have to interplay with environmental stressors. And that's why issues like workload, stressors, night shift works or substance use may precipitate new episodes. And the bipolar disorder working group of the PGC, we are focusing on discovering more of the genetic susceptibility, but we are really interested in understanding the whole picture of bipolar disorder. Thank you.
Hi, I'm Niamh Mullins, and on behalf of the Bipolar Disorder Working Group, I'll be presenting the results of the PGC3 Bipolar Disorder GWAS, and you can find the preprint of this paper on medRxiv. Firstly, just some brief background. Bipolar disorder is a severe psychiatric disorder characterized by unusual swings in mood, ranging from mania to depression in the case of type 1 bipolar disorder, or hypomania to depression in the case of type 2 bipolar disorder. Our previous GWAS, the PGC2 study, identified 30 genome-wide significant loci for bipolar disorder using a sample of approximately 20,000 cases. In the PGC3 GWAS, we have around 42,000 cases. Most of these data sets are internal to the PGC, and most of these samples are clinically ascertained using structured psychiatric interviews. We also have included 5 external biobanks, which mainly have ascertained cases using ICD codes or self-report measures. Our samples here include all of the bipolar disorder subtypes, and they're all of European descent. We identified 64 genome-wide significant loci, 33 of which are novel for bipolar disorder. These are shown here in yellow. We saw strong replication of loci from the PGC2 paper, with 28 out of those 30 replicating. And we also saw replication of the ODZ4 locus from the PGC1 paper and a few other loci from published GWAS. This is a closer look at the novel loci. Here they're labeled by the nearest gene to the index SNP. So the MHC is now genome-wide significant for bipolar disorder, and we were able to impute the C4 structural variants and predicted C4a expression levels in our data sets, and we found no evidence that the association here is driven by C4. Some other notable novel associations are the CACN-B2 calcium channel, KCNB1, which is a potassium channel, and furin, which is a gene with a neurodevelopmental role well known for its involvement in schizophrenia. We use MAGMA to look at signal enrichment in our GWAS results. Looking at gene sets, we found enrichment in 10 gene sets, all of which involve synaptic activity, neurogenesis, or calcium channel signaling. And looking at gene expression, we saw enrichment in genes expressed in all brain tissues from GTEx and no other tissue types, with the highest enrichment in the prefrontal cortex. In terms of the specific cell types involved in bipolar disorder, these are the results of cell-specific enrichment analyses from 5 human and mouse brain data sets. The biggest enrichment we see is in neurons in the cortex, but also in several subcortical regions, including both excitatory and inhibitory neurons. And in humans, we also found enrichment of signal in hippocampal pyramidal CA1 neurons and interneurons of the prefrontal cortex and hippocampus. These are the results of an enrichment analysis in the gene targets of different drug classes. We examined about 150 drug classes, and we found significant enrichment in 4 drug classes at the very broadest level. The top class was the psycholeptics, which are drugs that have a calming effect on behavior. And the most significant subclass of the psycholeptics were antipsychotics. This is a class that also includes the mood stabilizers. So it's reassuring that this method reliably detects enrichment in the targets of current bipolar disorder treatments. However, we also saw enrichment in the targets of calcium channel blockers, which are labeled for treating hypertension, drugs for treating GI disorders, which include a lot of anticholinergic and serotonergic drugs, and in the targets of antiepileptics. And calcium channel blockers and antiepileptics are sometimes used off-label as bipolar disorder treatments. So these results point towards new therapeutic leads and potential opportunities for repositioning some classes of drugs as bipolar disorder treatments. We've integrated eQTL data with our GWAS results in several ways in order to prioritize biologically relevant genes. Firstly, we use TWAS fusion and brain eQTL data from the PsychEncode Consortium. And from fusion, we found 77 genes for which bipolar disorder risk alleles significantly influence gene expression in the brain. And these 77 genes were clustered in 40 distinct genomic regions, which motivated us to fine map these regions. And this was done using Focus, which is a Bayesian TWAS fine mapping tool. And Focus found 32 genes with a posterior probability of greater than 0.7. And as a further sensitivity analysis, we then followed up on these 32 genes using SMR and data from the PsychEncode Consortium and also eQTLGen, which is the largest available eQTL panel of over 30,000 blood samples. So here we found 15 genes that were SMR significant and passed the Heidi test, which suggests the causal role of SNPs on bipolar disorder via gene expression. And we have some interesting genes here. For example, decreased expression of furin is associated with bipolar disorder as it is with schizophrenia. Reduced expression of DCLK3 was associated with bipolar disorder. And this is a gene near the TRANK1 locus, which is well known in bipolar disorder. It's actually the very top hit from our GWAS. However, our eQTL analyses found a stronger regulation of DCLK3 than TRANK1. We also have a couple of receptors here, HTR6 and MCHR1, which is the target of the antipsychotic haloperidol. So these are 15 promising candidate genes, which could now be followed up in functional genomic experiments to better understand their role in bipolar disorder. Polygenic risk scores from the new GWAS results explain on average 4.75% of variance in European samples. Looking in non-European samples, we saw about 2% variance explained in East Asian samples and less than 1% variance explained in admixed African American samples. And this is a big improvement on the results using the PGC2 GWAS summary statistics. However, clearly the performance is still lagging in non-European samples, and there's a need to increase the diversity in discovery GWAS of bipolar disorder. This slide shows the results of several analyses looking at the genetic and potentially causal relationships between bipolar disorder and some modifiable risk factors. So here we were interested in looking at sleep disturbances, mood instability, education, and substance use based on their relationship with bipolar disorder from clinical studies. And most of these showed a low positive genetic correlation with bipolar disorder. The Venn diagrams here show the results of Mixer, which shows the variance influencing bipolar disorder, specifically the variance influencing both bipolar disorder and the trait of interest, and unique to the trait of interest. So Mixer found that many genetic variants influence both bipolar disorder and all of our traits of interest, in particular educational attainment, where it estimated that almost all of the variants influencing BD also influence educational attainment. And the arrows above and below the Venn diagrams show the results of Mendelian randomization using GSMR and 2 sets of summary statistics. Here we found a significant effect of bipolar disorder on reduced likelihood of being a morning person, increased number of drinks per week. And looking in the opposite direction, we found that smoking conferred a 1.5-fold increased risk of bipolar disorder. And we found significant bidirectional relationships between bipolar disorder and several of the other traits that we looked at. So these sort of causal relationships could have potential clinical impact in the future in the prophylaxis or management of bipolar disorder. Although we consider this Mendelian randomization analysis to be exploratory, and these potential causal relationships would really need to be followed up in future experiments to fully understand their relationship with bipolar disorder. So to summarize, we found 64 genome-wide significant loci. If we look at the number of cases compared with the number of loci, it's fair to say that we've reached the inflection point now in GWAS of bipolar disorder, and we expect a lot of return on our increase in sample size going forward. We have found 15 promising candidate genes, which could be now followed up in functional genomic experiments to better understand their role in bipolar disorder. We've improved the performance of polygenic scores in both European and non-European samples, and we found enrichment of bipolar disorder associations in the targets of calcium channel blockers, anti-epileptics, and drugs for treating GI disorders, which suggest potential opportunities for repositioning some of these classes of drugs as bipolar disorder treatments. I'd like to thank all members of the Bipolar Disorder Working Group. Again, you can find the preprint of this paper on medRxiv, and here are the contact details for the corresponding authors, myself and Ule Andreassen, who is chair of the Bipolar Disorder Working Group.
Hi, my name is Kevin, and today I'll be presenting on behalf of the Bipolar Disorder Working Group of the PGC on some ongoing analysis of clinical versus population samples of bipolar disorder. I have nothing to declare. And so the current BD freeze includes about 40,000 cases and 370,000 controls. And we successfully identified 64 genome-wide significant loci, 33 of which are novel. And one of the successes of this GWAS is it seems like we've now reached the inflection point for discovery in bipolar disorder. As seen in the lower image here, we expect to follow the trajectory of schizophrenia, where we increase the number of significant loci with a lower increase in sample size required. And so as we increase with larger sample sizes, we expect more discoveries. Within this current freeze, the samples are also predominantly clinically ascertained, although samples such as the UK Biobank and Estonian Biobank have also now been included. And samples are also predominantly bipolar disorder type 1. So moving forward, one of the aims of the group is to increase discovery of bipolar disorder associated variants by analyzing larger and more diverse samples, and specifically to investigate differences in genetic architecture between clinical and population or self-reported samples. To do this, we've recently obtained data from 23andMe, which I'll refer to as a population sample moving forward. And in this sample, bipolar disorder was defined by a self-report. Cases answered yes to have you ever been diagnosed or treated with bipolar disorder, while controls answered no. And so from 23andMe, we received summary statistics of approximately 70,000 cases and 1.5 million controls. In this population sample, there are 76 genome-wide significant loci for self-reported bipolar disorder, of which 6 overlap with those discovered in the PGC BD freeze I mentioned earlier. When we meta-analyze the two samples together, it's approximately 114,000 cases, 1.9 million controls, all of European ancestry, and we identify 172 genome-wide significant loci, of which 138 are novel for bipolar disorder. We also identify a number of overlapping loci with other psychiatric disorders, including 7 for ADHD, 1 for autism spectrum disorders, 37 for depression, and 35 with schizophrenia. Investigating the genetic architecture in this meta-analysis, we identified a reduced genetic correlation with schizophrenia than what has been reported before, an increased correlation with MDD. And when we explored the two samples using bivariate mixer, we see that the genetic correlation between these samples is about 0.57, although they do share about 8,000 influencing variants, and the 23andMe sample seems to be more polygenic with an increased number of unique variants. So when we look at the two samples and compare their genetic correlations, we see that this drop in genetic correlation with schizophrenia in the meta-analysis is driven by the 23andMe sample, where we get a genetic correlation of only 0.34. And then conversely, the genetic correlations with depression, as well as ADHD and autism, increase in the 23andMe sample. Another interesting note is that when looking at the PGC2 bipolar disorder, which included more clinical samples than the current freeze, the genetic correlation is reduced for the overall sample, but is further reduced when we consider only bipolar type 1 disorder. So this prompted us to try to investigate or further investigate the difference between these phenotypes, bipolar 1 and bipolar 2 disorder. And so on the left here, we have done polygenic risk scoring into 3 independent samples of bipolar 1 disorder. The furthest left panel is the PGC3 data set. The middle panel is 23andMe data, and then the right-hand panel here is the meta-analysis. And so what we see is that the PGC sample seems to predict bipolar disorder type 1 more accurately than the 23andMe data set, although the meta-analysis does still increase in the power to predict bipolar disorder type 1. Conversely, when we look at bipolar disorder type 2, the PGC sample does a much poorer job of predicting bipolar disorder type 2 as compared to the 23andMe sample. And again, we see the best performance with the meta-analysis. So to conclude, our meta-analysis of this population-based sample from 23andMe and the clinical samples from the PGC yields 172 genome-wide significant loci, as well as numerous overlaps with other psychiatric disorders. And when we compared the population and clinical samples, we noticed a slightly different genetic architecture, where the clinical PGC samples seem to represent more bipolar disorder type 1 and are more strongly correlated with schizophrenia, whereas the population-based 23andMe sample appears to represent bipolar disorder type 2 and is more strongly correlated with depression. And so our conclusion is that the population sample may reflect the non-hospitalized and non-psychotic part of the bipolar disorder spectrum. And so in the next steps, these analyses are ongoing. And so we're looking for additional and new samples to include in our meta-analysis, as well as to analyze more subtype data and better characterize bipolar disorder type 1 and bipolar disorder type 2. At this point, I'd just like to acknowledge the working group, as well as the other groups we've collaborated with, as well as 23andMe, and please contact us if you have additional samples to include. Thank you.
Welcome, my name is Peter Zandi from the Johns Hopkins University, and I'll be presenting today for the Bipolar Sequencing Consortium. I'll be providing a very brief update on our investigations into the role of rare variants and the risk for bipolar disorder. The Bipolar Sequencing Consortium was formed over 5 years ago to investigate the role of rare variants in the risk for bipolar disorder. The goal was to bring together existing exome and whole genome sequencing studies of bipolar disorder that had been carried out around the world for combined analyses with both case control and family samples. The map shown here highlights countries where those samples had been collected. Most of the samples were collected from North America, Northern Europe, and Australia, although there were some samples from Central America, Iran, and Japan. In our first phase, we set out to carry out a meta-analysis of coding sequence data from 4 existing case control studies that had a combined sample size of just under 4,000 cases and over 5,300 controls. 3 of the samples provided exome sequence data, while 1 provided low-pass whole genome sequence data. Analyses of these data were led by Laura Scott at the University of Michigan and Sherman Gia, formerly at UCSF. We focused on testing for increased burden of rare pathogenic variants in certain gene sets that had been implicated in previous sequencing studies of schizophrenia, as well as had been hypothesized to be involved in bipolar disorder. We didn't find any evidence of increased burden of rare pathogenic variants across the exome, or in several synaptic gene sets, or gene sets of loss of function intolerant genes, which again had been previously implicated in schizophrenia. However, we did observe evidence of increased burden of rare pathogenic variants in genes that had been previously implicated by the PGC GWAS studies of bipolar disorder. These findings raise the intriguing hypothesis that genes that harbor common variants for bipolar disorder may also harbor rare risk variants. We sought to replicate these findings in an independent sample that was being gathered by the Bipolar Exome Sequencing Study at the Broad Institute under the leadership of Ben Neal. At the time of replication, they had collected data on just about 10,000 cases and 14,000 controls. Unfortunately, we were unable to replicate the finding of increased burden of rare pathogenic variants in these bipolar GWAS sets across any of the samples that had been brought together for the BIPEC study, and regardless of how we defined these bipolar GWAS gene sets. We sought to examine whether there were any differences between the samples in the replication and the initial samples that we studied that might explain the difference, but couldn't find any. We concluded that there was insufficient evidence to suggest the role of rare variants in the risk for bipolar disorder, at least in the gene sets that we tested. This stood in contrast to findings from a previous sequencing study of schizophrenia, which did find evidence of increased burden of rare variants in these gene sets with a smaller sample size than what we had gathered. However, since then, the BIPEC study has gathered an even larger sample of cases and controls across Europe and the USA. These samples were all exome sequenced at the Broad Institute and jointly called in a common pipeline. After all QC procedures, they had a sample size of just over 14,000 cases and 14,000 controls. Analyses of these data were led by Duncan Palmer, again, under the guidance of Ben Neal at the Broad Institute. And finally, with the increased family size, we started to see the first evidence of a role of rare variants in bipolar disorder. They found a significant increased burden of these ultra-rare protein truncating variants, specifically in loss-of-function intolerant genes, as shown here. The significant increased burden was observed both in bipolar 1 disorder samples as well as bipolar 2 disorder samples. There wasn't a similar increased burden of variants for missense variants or synonymous variants. However, there was a hint of increased burden in damaging missense variants in these particular loss-of-function intolerant genes, but these findings are unclear and larger sample sizes will be needed to determine whether there's anything there. In addition, they tested for increased burden of these rare protein truncating variants in individual genes, as shown here in this Manhattan plot. None of the genes were significant after controlling for multiple testing, the threshold of which is shown here by the dashed red line. However, they did see a deviation in the signal of association from the null expectation. And in fact, when they combined this data with data from Schema, which is a parallel consortium effort to combine sequencing data in schizophrenia, they did observe that several of these genes were significantly associated, even after correcting for multiple testing. These findings highlighted that there were certain genes with risk variants that were in common between both bipolar disorder and schizophrenia, echoing the findings we are observing with common variants from GWAS of an overlap in genetic risk for bipolar disorder and schizophrenia. In addition, upon further investigation, they found that these top associated genes were enriched in genes that are expressed in CNS tissue. In particular, they found an enrichment in genes from the amygdala. This is interesting, perhaps because the amygdala is part of the limbic system, which has been hypothesized to be involved in mood disorders. They also tested a number of gene sets for increased burden of these variants, and they did find evidence of increased burden of gene sets related to schizophrenia, in particular, a gene set consisting of those genes that were found in the Schema Consortium finding, again, highlighting the overlap in rare variant risk between bipolar disorder and schizophrenia. Together, these findings are very exciting and are finally beginning to illuminate the potential role of rare variant risks in bipolar disorder. At the same time, we've been analyzing data from our family samples to see what we can learn from them about the contribution of rare variants to the risk for bipolar disorder. This work has been particularly challenging because of the heterogeneity in the samples that have been brought together in regards to their family sizes that have been studied, the population background, and the sequencing platforms. For example, several studies were of families from the Amish population, and they involved a few number of families, but with very large number of affected relatives. In contrast, other samples involved samples from more general population with larger number of families, but a smaller number of affected relatives. There was a fair mix of data from both exome sequence data and whole genome sequence data from a number of different platforms. We spent a fair bit of effort to try to harmonize this data so that we could examine it and test it for rare variant segregation, as well as for increased polygenic risk scores to see if that could explain the segregation of disorder in these families. Analyses of these data are being led by Fernando Goes at Johns Hopkins University and Rachel Kember at the University of Pennsylvania. After extensive QC, We had data for analysis on 234 families with 2 or more affected relatives, and a total of 834 affected relatives across each of these families. We observed over 15,000 rare variants fully segregating in 1 or more of these families. When we looked at more deleterious variants with a CAD score greater than 15, we observed over 7,000 such variants, and looking at disruptive variants, over 500 such variants. When we focused on those variants with a CAD score greater than 15, shown over here, we observed a total of 1,445 genes that had variants that were fully segregating in 2 or more families. Some of these genes had multiple variants segregating in multiple families, such as SIGN1. SIGN1 is a very interesting candidate gene. However, it's also a very large gene like Titin, shown here. We are currently further analyzing this data to test whether the observed segregation is greater than what we would expect by chance given the pedigree structures and the sizes of these genes. We're also testing to see whether the genes where we do observe these segregation events are enriched for genes that have been identified in the case control studies that we just described. We've been also interested to test the idea whether these families have an increased burden of polygenic risk scores, as had been suggested by several previous family studies with smaller sample sizes. To do this, we combined data from our family samples with data from a case control sample and imputed the data to over 5 million common variants with the top med reference panel. We then calculated polygenic risk scores for both bipolar disorder and schizophrenia after removing any overlapping samples. The results for bipolar disorder are shown on the left, for schizophrenia on the right. As expected, we observed a higher polygenic risk score among cases in our case control sample compared to the controls. Interestingly, we observed an even higher polygenic risk score among our affected relatives in the families and a higher polygenic risk score in our unaffected relatives. You see the same pattern here with schizophrenia. We are currently testing whether the observed increased polygenic risk scores in the families are statistically significant, and we are testing whether the polygenic risk scores seem to be interacting with rare variants that are segregating in these families. This work is ongoing, and we hope to be able to report and publish on these results within the year. Taken together, these findings are very encouraging, especially with regard to the case control samples. They're beginning to illuminate the role of rare variants in the risk for bipolar disorder, but much remains to be learned. Clearly, we will need larger samples if we want to make progress in this regard. There are several large-scale efforts ongoing that may be able to help with this effort, including the All of Us study, the collaboration between Geisinger and Regeneron, and the VA MVP program. Each of these efforts are collecting large numbers of cases and controls and sequencing them, and there will be a fair number of bipolar cases within those samples that may be able to contribute. In addition, it would be great to include additional sample collections specifically with bipolar disorder for these sequencing studies. We also need more diverse samples, as can be seen by the map, which we're showing again here. Samples from South America and Africa and Asia are clearly underrepresented or non-existent. Finally, we will need better phenotyping on these data. It has been hypothesized that the extensive clinical heterogeneity that we observe in bipolar disorder may reflect the underlying genetic heterogeneity, and this may be particularly true for the contribution of rare risk variants. We are currently contributing to an effort being led by Nelson Freimer and Annabel Vreeker to pull together the clinical phenotype data from these samples with sequence data from around the world so we can investigate this further and to see if it can help us further clarify the role of rare variants in the risk for bipolar disorder. It would be great when we collect larger samples, we also collect additional phenotyping data to help in this effort. Finally, I'd like to thank all the investigators from around the world who have contributed to this very challenging and extensive effort. I'd also like to thank the support from the NIMH and the Dalio Foundation for this work. It really does take a village to make progress, and thanks go to the community. Thank you for listening.
My name is Annabel Vreeker, and I'm a postdoctoral researcher at the Erasmus MC in Rotterdam, the Netherlands. Today I will tell you something, also on behalf of Ariana Di Florio and Andrew McQuillan, on the importance of deep phenotyping in genetic studies. I will give a brief overview of several efforts that are currently being conducted by different consortia, and I will also show you the results of 3 projects that have been recently published or submitted. So first, why deep phenotyping in psychiatric disorders? We know that psychiatric disorders are largely heterogeneous and that there are many overlapping symptoms across disorders. In addition, there's substantial genetic overlap between disorders. So in order to understand the biological mechanisms underlying psychiatric disorders, it is essential to not only study the relationship between diagnosis, but also of subphenotypes with genetic variation. Over the past years, several large-scale genetic consortia have focused on subphenotypes in relation to genetic variation, and this is also supported by the NIMH. In the PGC, the Whole Genome Sequencing for Psychiatric Disorders Consortium, and the Bipolar Sequencing Consortium, we have collected and analyzed subphenotype data of patients with available genetic data. And we have collaborated also with other large-scale consortia, such as Schema and PGC Targeted Sequencing Group. The subphenotype data that we have access to mostly come from multiple psychiatric instruments. And because those psychiatric instruments are not completely the same, we have to account for differences that occur across those instruments. Within the WGSPD, Max Mansell, Bob Bilder, Stephen Rees, Nelson Freimer, many others, and myself have worked on statistical models to generate harmonized dimensional phenotypes from thousands of patients with data from multiple psychiatric instruments. And we have generated factor scores for hallucinations, delusions, depression, mania, disorganized symptoms, and negative symptoms. And we have published on this method in Educational Psychological Measurement. Within WGSPD and Bipolar Sequencing Consortium, we are currently also working on an effort to analyze item-level data of over 50 items on mood symptoms and psychotic symptoms in relation to genetic variation. Last, there have also been efforts, primarily within the PGC, to study disease characteristics in relation to genetic variation. And I'm highlighting here 3 projects on bipolar subtype, age and polarity at onset, and sleep. I want to emphasize that this is just an initial set of phenotypes. Within the PGC, there is interest in many more phenotypes, such as substance use, comorbid disorders, and suicidality. But I'm focusing on these 3 projects because I want to tell something more about the results of these projects. So the first project is on bipolar subtypes, and this is a study that was published last year and was led by Eli Stahl. In this GWAS, this was a GWAS on subtypes of bipolar disorder, including almost 15,000 bipolar 1 cases, over 3,000 bipolar 2 cases, and almost 1,000 schizoaffective cases. Within bipolar 1 cases, there were 14 genome-wide significant loci identified. And heritability analyses showed that heritability estimates were higher for bipolar 1 and schizoaffective cases as compared to bipolar 2. An interesting finding was that the genetic correlation of schizophrenia was stronger for bipolar 1 as compared to bipolar 2. In contrast, the genetic correlation of depression with bipolar 2 was stronger as compared to bipolar 1. And this distinction between schizophrenia genetic liability and depression genetic liability was also shown in polygenic risk scores analyses. They found that schizophrenia polygenic risk scores were higher for bipolar 1 cases as compared to bipolar 2 cases. And that depression polygenic risk scores were higher for bipolar 2 cases as compared to bipolar 1 cases. The second project is on sleep, and this is a project that was carried out by Katie Lewis, Ariana Di Florio, and many others. And in this project, they focused on the comparison of genetic liability for sleep traits among individuals with bipolar 1 or 2 disorder and control participants. They carried out this study because several studies in the general population have identified genetic loci to be associated with sleep traits such as insomnia, sleep duration, chronotype. And we know that these sleep traits are also associated to bipolar disorder, and that bipolar patients can also experience disturbances in these sleep traits. So they carried out a study in which they investigated the association between genetic liability of sleep traits with bipolar 1 and bipolar 2 disorder. And they particularly investigated whether there was a difference in the association between subtypes. For this study, they used the BDRN cohort, a large UK cohort, including over 10,000 individuals. And they also used a replication cohort from Sweden, including over 10,000 individuals. And indeed, they show that genetic liability differs for bipolar 1 and bipolar 2 cases. In particular, polygenic risk scores for insomnia were significantly associated with bipolar 2, but not with bipolar 1. Polygenic risk scores for sleep duration were significantly associated with bipolar 1, but not bipolar 2. And they also found these associations in the Swedish sample. They did not find a significant association between polygenic risk scores and daytime sleepiness and chronotype with bipolar subtype. The third project that I want to talk about is on age and polarity at bipolar onset. And I've been working on this project with Janos Kalman, Loes Olde-Lohuis, Til Antlauer, and many others. In this project, we aim to characterize age and polarity of bipolar onset in almost 13,000 bipolar patients. And the reason that we wanted to do this is that we know that age and polarity of bipolar onset are important predictors of disease scores, but the underlying nature remained largely unknown. So we set out a study in which we examined phenotype definitions and associations with disease characteristics. We conducted systematic GWASs, and we investigated the association of polygenic risk scores for neuropsychiatric disorders and traits and age and polarity of bipolar onset. And as illustrated in this figure, we found systematic differences in onset phenotypes between cohorts and continents of origin. In addition, we found one significant locus in the age at onset GWAS, but we could not replicate this finding. In polygenic risk score analysis, we found significant associations of polygenic risk scores for ASD, MDD, schizophrenia, and educational attainment with age at onset. However, we found no significant associations with polarity at onset. An important finding in our study was that when we calculated heritability estimates, we found that heritability estimates decrease when less strictly defined phenotype definitions were used and when different phenotype definitions and continents were combined. And this is an important finding because this really underscores the influence of phenotypic heterogeneity in genetic analyses. And it indicates that a heterogeneous phenotype can reduce power in genetic analyses. So in conclusion, I showed that there are differences in biological mechanisms underlying different phenotypes of bipolar disorder, which shows the need for deep phenotyping in genetic studies. I showed that phenotypic heterogeneity can lead to reduced power for genetic analyses, emphasizing the need for harmonized methods to assess phenotypes. There are several ongoing phenotype efforts by the PGC, the WGSPD, and the Bipolar Sequencing Consortium, and also other consortia. And it's important that we collaborate as much as we can, which we do, because we really need large sample sizes for these kind of studies. I would like to thank the NIMH, all the participants, the PGC Bipolar Working Group, the WGSPD Consortium, the Bipolar Sequencing Consortium, Roel Ophoff, Thomas Schulze, Janos Kalman, Til Antlauer, Loes Olde-Lohuis, Nelson Freimer, and all the other people that are listed here and who have contributed to our phenotype efforts. And I would like to thank you for your attention.
Hi, my name is Carlos Lopezjaramillo. I'm professor of psychiatry at the Universidad de Antioquia in Colombia. Today, we are going to discuss some of the, and show you some of the projects that we are doing in a very interesting population here in Colombia. This population is in the state of Antioquia in Colombia, and you can see in the map as the northwestern region of the Colombia is in the northwestern region of the continent, and this state Antioquia is in the mountain region in Colombia. This special population is called the Paisa population, and you can see in the map the area that is distributed this population in Colombia. What is the special of this population? This population is like a very interesting characteristics. It's very common to have a very extensive families. We say in the nuclear family, we can have family with 20, even more members, and also very religious, Catholic religions in this area, in this population, with interesting data about the high prevalence of the severe mental illness in this population. You can see we had in small towns high prevalence of bipolar 1, like around 6-8% of the population. And also in some areas, we can find the suicide rate higher than the rest of the country, with 10 times higher than the rest of the country and the rest of the areas in Colombia. So for that reason, for more than 20 years, we start looking this population, and very interesting because in the Central Valley of Costa Rica, there's a very similar and very homogeneous founder population with a very similar characteristics that this Paisa population. So for that reason, we study interesting studying bipolar disorder, bipolar 1 in large pedigree with, you can see more than 1,000 members, with around 200 of them with bipolar 1. So we did this study with looking different phenotypes in this bipolar 1 population, and we found in this population interesting phenotypes related with the bipolar 1. Phenotyping, like even in neuroimaging phenotypes, like particularly the cortical thickness in prefrontal and temporal regions associated to the bipolar 1 in this population. When we are doing this study with the Central Valley of Costa Rica and also the Antioquia pedigrees, we also, see that there's a lot of, we find in these families a lot of prevalence, a higher prevalence of different disorder like psychosis, depression, severe depression, etc. So for that reason, we decide to, to start looking in this, specific in this Paisa population here in Colombia, to look in the focus not only in one disorder like a bipolar 1, like the previous study that we did with Costa Rica. And so in this one, we start this is a current project, and we start with this one like almost 3 years ago, looking for the subjects with major depression, with severe major depression, with bipolar disorder and schizophrenia. So the idea is we're gonna have 8,000 cases and 2,000 controls in this population, looking different level of diagnosis, focus on specific symptoms and quantitative traits. For that reason, the procedure that we are doing is as soon as we get the contact, the screening, the social worker, and the subject that we're going to include in this study. We are doing the neuropsychological assessment with the pen battery, and also the psychiatric assessment with the NEST kit, and focus, like I said before, in the diagnosis of psychosis, bipolar, and major depression. And that is the the preliminary results that we had right now. We have already assessed more than 4,000 subjects, and you can see in this table, almost 1,000 subjects with bipolar 1, more than 300 with bipolar 2, and almost 2,000 subjects with major depression. And also the age, the mean age is 43 years old, very similar to the subjects and controls. And we already have some preliminary results in this population. And like I told you, this is a current project. The idea is to finish with all these projects in 2 years. So that's the preliminary result that we have related with the cognitive performance in this population. And it's interesting that already we can see like 2 big groups. One is subject with bipolar 1 and schizophrenia subjects with similar impairment in accuracy and speed across cognitive domains. And in the other side, we have subject with bipolar 2 and major depressive disorder with similar performance, also with more deficit in executive and social cognition. So it's like 2 different big groups in the cognitive performance in the subjects. And the idea is to start to looking, markers associated to the big groups and see if there is a specific marker related with the cognitive performance in this population. So that is the results that we are already have, and the idea is to try to understand more the causes and trajectories of the severe mental illness. So, but when we start looking, after we are looking the data that we found in this population during the interview, but also we have the access to review the electronic medical records, we found that in the electronic medical records, we can find a very, very interesting information, even more, clear and even more important information than a single interview. So for that reason, we applied for the idea is that this year, we're gonna start this Latin America biobank for late-scale in genetic research of severe mental illness. So the idea is to, we are going to look at in 50,000 cases with severe mental illness. Again, schizophrenia, bipolar, and severe depression, and also 50,000 controls. So now we are doing only, we're gonna have a very short interview. Is to have the informed consent, and we planning to review and also made up that, the idea is to make up data analysis of the electronic medical records in the 50,000 cases, and also to see what we can, find in these big numbers related with the, try to see the correlation between genes and the information that we can, that we're gonna find in the electronic medical records. Well, this is the you know, the project that we're gonna start very soon in Colombia in this Paisa population. But also, is the idea is to try to, you know, in that very different strategy to looking in different ancestry. So in this case in Latin America and also in the African ancestry. So for that reason, we are going to be part of this very important project, the Pumas Project. The idea is to build a bank of the cases with schizophrenia, bipolar, and controls in not only from Africa, also from a mixed population in this case in Latin America. So we're gonna have a very interesting population and with a very similar strategy, looking the electronic medical records and also a short interview. In this case, we're gonna use the DPAD for the interview, and the idea is to start with a mixed population in Colombia and Brazil, and that is the Latin part of this big project, and in a very different strategy. You know, in the Paisa and this homogeneous population, you know, the idea is to focus in the very mixed population in Colombia, in the Caribbean coast, and also in Brazil, with areas with more mixed population and with the African ancestry. So this is the project that we hope that we can start to the assessment this year, yeah, and in Brazil and Colombia. So like you can see in this short time, I'm going to, you know, only have show you the projects that we are developed here in Colombia, the Paisa project, the Biobank, the Paisa Biobank, and also the Pumas. So for that reason, I hope that in few years we're gonna have that very, very interesting information with an important number of subjects and a big, you know, opportunity. We have a lot of opportunity to explore and looking in different areas, and not only focus in this severe mental illness, maybe in different, even in different disorders, not only in psychiatric disorder. So I hope that in few years, like I tell you, we can share important information about these projects. So this is a picture of the team from UCLA, UPenn, UCSF, Rutgers University, and my team from Colombia, Universidad de Antioquia, and Universidad de Caldas. So thank you very much.
Hello, my name is Ming Li. I'm from Chinese Academy of Science. And today I'm very happy to be here, and I would like to share with you some of our recent findings in a bipolar disorder GOS in Han Chinese population. Bipolar disorder has been recognized as a severe mental illness in most countries of the world, including China. However, this disorder still remains underdiagnosed due to many reasons, and its biological basis is still relatively unclear. Nevertheless, due to the high heritability of bipolar disorder, suggests the potential roles of genetic factors in its pathogenesis, therefore calling for the large-scale genetic association studies to identify the credible risk variants. So far, multiple genome-wide association studies with collaborative efforts to achieve a giant sample sizes have been conducted in the populations of European ancestries. While there is only one bipolar disorder GOS in Chinese population, which was published on 2011. However, these studies did not identify any genome-wide significant loci. Our group in collaboration with multiple mental health centers in China, have collected a relatively higher, larger bipolar disorder case control sample, and all the cases were diagnosed based on the DSM-4 criteria, and all the controls were recruited from local communities based on their self-reported as mentally healthy with their with no personal or family history of mental illness. And of the individuals were Han Chinese origin, and after a strict quality control, the sample size included in the final analysis were over like 6,000. Before our case control analysis, we firstly performed a principal component analysis, and here are the PCA plots as well as a QQ plot of our discovery jewel samples, and we also show that the LD score regression analysis, and here is the intercept and the attenuation ratio. As you can see from this results, there is a negligible population stratification in our GWAS samples. And in addition, based on our SNP heritability, based on the LD score regression, we observe a relatively high SNP heritability in our GWAS samples. And here is a Manhattan plot for our GWAS, Han Chinese discovery GWAS. In brief, we have identified a SNP located near the TMEM108 gene, showing genome-wide significant association with bipolar disorder. And this SNP also shows nominal association in an independent Han Chinese replication sample. However, it is also notable that compared with the previous GWAS in Europeans, the Han Chinese GWAS sample size is still much smaller. And here is a Lucas-Toom plot for this genome-wide risk locus. From this plot, we can see that there are two genes near the risk variant. The first gene is the NPHP3 AS1, and the other gene is the TMEM108. The NPHP3 AS1 is a non-coding RNA, and based on the RNA-seq data from the GTEx and the brain span data sets, this non-coding RNA is expressed is rarely expressed in most human organs, including the brain tissues. However, the TMEM108 is abundantly expressed in human brain tissues. More importantly, based on previous studies, the protein encoded by the TMEM108 is enriched in the post-synaptic density fraction, and the deficiency of this gene in neurons results in smaller and fewer spines compared with the wild-type neurons, and the TMEM108 mutant mice have decreased the excitatory post-synaptic currents and the adult neurogenesis of the hippocampal DG neurons. And for the behavior tests, the deficient mice of this gene displayed the compromised sensory motor gating, the cognitive function, and the lower immobility in tail suspension test, and the form swimming test compared with the wild-type mice. All of these studies have confirmed that this gene plays important roles in neurodevelopment and the synaptic-related biological processes. And we believe that these results have implicated the potential biological mechanisms underlying the risk associations with the bipolar disorder. After our primary GWAS analysis, we have conducted the genetic correlation analysis between our Han Chinese bipolar GWAS and the other psychiatric disorders and traits using the popcorn and the LD score regression analysis. First of all, through the analysis using the popcorn, we have shown that there is a strong genetic correlation between our Han Chinese bipolar GOS and the European PGC2 bipolar GOS. By performing both the LD score regression and the popcorn analysis, we also show that our Han Chinese bipolar GOS has a strong positive genetic correlation with schizophrenia, both in East Asian and in European populations. And from the popcorn analysis, we also found that our Han Chinese bipolar GOS also shows strong negative genetic correlations with cognitive performance, intelligence, and educational attainment. In addition, we also show that we also noticed that the significance of genetic correlation between bipolar disorder and the depression is much lower than that of the correlation between bipolar and the schizophrenia, which is also consistent with the previous studies. After the genetic correlations, we have also conducted the polygenic risk score analysis using the same data sets, and the results are generally consistent with the aforementioned genetic correlations. And here are the primary conclusions based on the PRS analysis. In summary, the PRS analysis, the popcorn, and the LD score regression analysis have shown that the Han Chinese and the European bipolar have shared substantial genetic basis, and we therefore performed a trans-ancestry meta-analysis using our Han Chinese bipolar GOS and the European PGC2 bipolar GOS. And this trans-ancestry meta-analysis has identified two novel genome-wide significant loci, which were not reported before. The first one is near the VRK2, and the second one is located in the RHEBR1 gene. In addition, the previously reported GOS loci, such as the CACN1C, the NK3, TRNK1, HDC5, and the others were also genome-wide significant in our meta-analysis. Overall, we believe that the trans-ancestry meta-analysis have increased the resolution for the mapping analysis in bipolar disorder. These are the primary results of our recent GOS analysis of bipolar disorder in Han Chinese population. We are sincerely grateful for the contributions from all participating research groups, and we also very appreciate the excellent advice and valuable insights from the anonymous referees during the peer review process. And thanks for your time and attention. And I'm very happy to answer any questions.